Hey guys, welcome to APs with Adam. Today we will be covering the main ideas and overview of Chapter 17 in AP Biology. This is going to cover the differences between DNA and RNA, specific functions of RNA, how transcription occurs, and translation. Let's get into it. Let's begin with the differences of DNA and RNA. So as you can see here, the sugar in RNA is ribose. Now, deoxyribose in DNA is a ribose sugar, but with one less oxygen than in RNA. So in DNA, the bases are A, C, T, and G, and the A and the T, obviously, and the C and the G. Now in RNA, it's going to be A, and then instead of T, it'll be U. So if you see in DNA, sorry, in RNA transcription, if you see a T on the all right, let's begin with the differences of DNA and RNA. So as you can see here, the sugar in RNA is ribose. Now in DNA, it's deoxyribonucleic acid, which just means that there's one less oxygen than in the ribose sugar. So the base, that's one difference, is the base sugar is going to be ribose in RNA and deoxyribose in DNA. In DNA, the bases are A, T, C, and G, whereas in RNA, it's going to be A, U instead of T, and then C and G. And another difference is DNA is double-stranded, whereas RNA is only going to be single-stranded. DNA, they also have like different functions. So DNA, if you guys already know, is more like the cookbook in the cell. It has all the recipes, whereas RNA is going to be the specific, or mRNA is going to be like the specific recipe, and then the other RNA are just going to try to create that recipe that was just transcribed. All right, so let's talk about the specific RNAs. All right, guys, so let's talk about the specific types of RNA. So I was talking about messenger RNA, how that's like more like the specific recipe. So as you can see here, it's only going to be one strand, like we said. There's not going to be the T. Instead of thymine, it's actually going to be uracil. And yeah, so we'll talk about the transcription process in a second. But ribosomal RNA is going to be composed of two subunits. There's going to be the small subunit on the bottom, and then the other subunit. It almost looks like a burger or burger buns, and then the mRNA, which is right here, will form in the middle here, acting as like the burger, if you think of it like that. Now, tRNA, this is going to be really important in translation. So as you can see here, there's an amino acid on it. The amino acid is going to be added to the existing peptide chain that we're going to be building up. We'll get into the specifics of that later. And then this right here is going to be called the anticodon. All right, guys, let's talk about transcription. So transcription is going to look very similar to DNA replication. So the first thing that's going to need to happen is the opening up of that DNA molecule. So as you can see here, this DNA strand is going to open up. Now, this looks very similar to DNA replication because it's a very similar process. Now, instead of in DNA replication where both sides will be replicated, only one side is going to be only going to be transcribed. So what we're going to see is the actual strand that is being transcribed is going to be called the template strand or the coding strand. Yeah. So the RNA, with the help of transcription factors, is going to bind on, and it's just going to build a continuous strand. And keep in mind, RNA does not need any primer, so it can just build. All right, so guys, just keep in mind, since the bases on RNA are A, U, C, and G, whenever an A on the DNA is seen, the complementary base will be U. All right, so once we've created that mRNA molecule, which is obviously complementary to the DNA, meaning that it's the DNA that was being used as the template is just going to pair up with that. It's not going to be exactly like that. Now, we in that process we create something called a pre-mRNA strand. Now, what that simply means is that it has some junk portions on it, which are called introns. So introns are segments that don't really code for anything, and the only reason why they're there is to decrease the risk of errors happening. So what's going to happen is these introns will be identified, and they'll be cut out by an enzyme called spliceosomes. Now, once we get rid of the introns, so once we're left with all exons, which are the coding segments, we're going to need to attach something called a 5-carbon cap, which is obviously going to it's going to attach to the 5-carbon end of the mRNA, and the poly A tail will add to the 3-carbon end. Now, the purpose of these two things are so that the mRNA molecule that we just spent a lot of energy making and resources is going to be safe once it leaves the nucleus to go to the cytoplasm to produce the proteins. So just keep in mind, this is only in eukaryotes. Prokaryotes do not have these the introns. All right, and one last thing. So sometimes the RNA will will purposely be shuffled. This is called alternative splicing. So what will happen is we can remove all the introns, that's a given. We're going to get rid of all the introns, but sometimes we might remove one of these, and we'll have a different protein. So this just allows us to have more options for proteins. All right, so this mRNA strand will move into the cytoplasm where the amino acids are present. The mRNA strand has segments consisting of 3 bases that code for a specific amino acid. So keep in mind, guys, mRNA is read in the 5 to 3 direction, whereas DNA is in the 3 to 5. All right, so in the cytoplasm, once the mRNA has entered there, a ribosomal subunit, which is made out of RNA, as I said, will position itself on the bottom portion. Now, the large subunit will bind on top of the mRNA and on top of the small subunit. This will create the ribosomal complex, which looks very similar to a burger, like I said. The rRNA complex or the ribosomal complex has 3 binding sites for the transfer RNA. So what's going to happen is there's the A site. Think A for adds. The tRNA is going to bind here and add this amino acid that it has, let's say it's in there, to the existing one. Now, so the A site is where the tRNA enters and then adds its amino acid, like I said. The P site, which holds together the amino acid chain, as well as the E site, which is where the tRNA will exit. So the energy source will be GTP. And once the ribosome has binded on the mRNA strand, it will search for the start codon. So before this complex occurs, it'll be like the bottom subunit will bind, then the top one, and what's going to happen is they'll form that complex and then search for AUG, which is the start codon. All right, so the tRNA here is going to have this thing right here, which is called the anticodon. Now, the anticodon is going to have the opposite of what is listed in these codons right over here. Now the reason this is so that it can bind in the right place and add its amino acid, that it's going to add this amino acid. But in order to bind there, it just has to have the opposite. So the opposite of this, what codes for this amino acid, is this. And the way that the amino acid gets its actual, the tRNA gets its amino acid, there's an enzyme called aminoacetyl tRNA synthetase. This is just going to add the correct amino acid from the cytoplasm onto the tRNA. This translation, what we just produced, that polypeptide chain, is only the first stage of the primary stage of protein production. So what we're going to have to do is, in the secondary stage, the folding of the amino acid chain will occur into alpha helices or beta sheets. In the tertiary stage, the R groups will interact, forming H bonds and further fold more. And finally, in the quaternary structure, which will combine multiple polypeptide chains. Keep in mind that the quaternary stage is not required to build a protein, because after the third or tertiary stage, a complete protein is produced. Point mutations are different than chromosomal mutations, as we covered in Unit 5. So there's going to be different types of point mutations. So a point mutation is just where one specific letter is swapped out, or base. So on DNA, it's always going to happen from the DNA, because that's like the starting point. So if a T is swapped out, it's just going to affect the whole thing. Now there's different types. So sometimes a silent point mutation is when there is no change in the amino acid that's produced. For example, here, even though the third T was swapped in DNA, and therefore which affected the A, this amino acid was still produced, which is the same as if it weren't to be swapped. Now, in nonsense mutations, a letter is swapped out or substituted, and it codes early for a stop codon. Now, the stop codon is just going to end the peptide chain production, and it's going to make it's not going to be able to make a protein. Now, the missense is when a different type of a different amino acid is also is just when a different amino acid is coded for, and it changes the function of the protein, but it still is a peptide chain and functional, just not in the function that the cell is trying to have. Frame shift mutations are very simple. So instead of being substituted, a base will be either added or deleted. So as you can see here, if the original one was the fat cat sat, and the T was removed, everything's going to shift to the left one. Now this, basically the cells are going off of this to code for the amino acids, what we call codons, as I said. So because everything shifted to the left, this is just going to code for something entirely different. This is an example of a deletion. Now, if we have an addition, so if something else was added, everything would be shifted to the right. And that would also have a similar effect, just messing up all the codons. All right, guys, so I hope you enjoyed that video. Make sure to leave a like, consider subscribing,